Hello, I'm Bevan and I'm one of the scientists at Essen BioScience, and this is our poster quantification of live cell imaging assays for immunotherapy, mainly looking at chemotaxis, immune cell killing and phagocytosis. These are three assays amongst the suite of assays that we are offering to cover the immuno-oncology applications. All of the assays follow a similar workflow that can be seen down this side of the poster. They're all based around our instrument, the IncuCyte ZOOM, and they're all looking at live cell assays that are looking at real-time measurements within the IncuCyte ZOOM machine. They are simple mix and read assays with no wash, no lifting and no fixing of the cells. The instrument itself provides you with an automated image analysis and image capture system, and what we're looking at here is time-lapse images taken from every single well and then inline automated analysis of those images. So all of the assays are non-perturbing, non-invasive and are direct measurements of the phenotypic cell biology that we're interested in. The machine has the capability of collecting high-definition phase contrast images, but also fluorescent object process analysis. The machine will provide you with real-time quantification of your biology, and here is a representation of a plate readout from the system. And that data can then be converted into time courses of your biology and then obviously kinetic readouts for that biology that you're looking at. And this is provided in an automated analysis package. So the three assays that we're particularly interested in on this poster, the first one here is the chemotaxis, and this is directional cell migration. And to use this application on the Zoom, we've created a new plate, which is the IncuCyte Clearview cell migration plate. And what you have here is an upper chamber and a lower chamber, which is separated by a membrane that has pores through it. And by the placement of the chemoattractant in the bottom reservoir, we can track the movement of cells from the top side through the pore and onto the bottom side here. And the instrument itself can take an image from the top side of the membrane and the bottom side of the membrane to allow you analysis of both sides. So the plate itself is amenable for use with adherent and non-adherent cell types, and can also be run as an invasion or a trans-endothelial migration assay. The images down here just show you a cell that is moving across the surface here towards the pore, which is circled here in orange, towards the chemoattractant. And as the time goes along here, you can see the cell is moving into the pore and then eventually appears on the underside. So these particular images are taken from the top side. So the cells that are nicely in focus here are cells on the top side of the membrane, and the cells which are a little bit more blurred here are actually cells on the bottom side of the membrane. But the integrated analysis will allow you to actually mask the cells on the top surface, and then if we flip to look at the image on the bottom surface, you can mask the cells on the bottom surface and therefore provide a count of both the top and the bottom surface. The data highlighted here just shows you some example data with, in this case, adherent cells. So these are human blood-derived macrophages moving in response to C5a. And what we're looking here is the images from the bottom side of the membrane, and we're actually looking at increase in cell area on the bottom side in response to the C5a in the bottom reservoir. Without the chemoattractant, we can see there are no cells that have come through to the bottom side. The other graph here is actually an example with neutrophils, so these are a non-adherent cell type. And in this readout, the cells are on the top side of the membrane, but once they move through the pore, they drop from the underside into the reservoir plate. So what we're actually measuring here is movement towards FMLP, but we're looking at a loss of area from the top side. And without the chemoattractant in the bottom well, you can see the cells just remain on the top surface. So using this particular assay readout, we've then gone on to look at some primary T-cell pharmacology. And what you can see here is a plate out view, which has come directly from the IncuCyte ZOOM software. And you can very quickly see that there are dose-related or concentration-related effects with the two ligands that we're looking at here. One is CXCR11 and one is SDF1 alpha. And in this case, the primary T cells are moving towards these chemoattractants. If we now convert the data here into the time course data that's shown here, you can see there's a nice concentration-related effect in the movement. And again, in this instance, the cells are going to drop off from the underside and we're looking at a loss of area over time for these cells. By then converting this to a CRC, we can look at the area under these curves and then get a nice CRC plot. The last bit of data here is actually looking at transendothelial migration data. And in this case, again, we're looking at primary T cells, but they're now moving in response to SDF-alpha, but across a monolayer of HUVEC cells. So they're actually having to be pulled through the endothelial layer. And again, we can see a nice time course here, and then we can see this converted into a CRC. The next set of assay that we're going to look at here is immune cell killing. And this particular assay setup is looking at a cold culture of tumour cells and immune cells. And in the pictorial here, you can see the tumour cells have actually got a red fluorescent nuclear label in them. This is just to aid us being able to track the proliferation of the target cells in the assay. The other component that we would have added into here is actually a caspase 3,7 apoptosis reagent. And what this will do is when the immune cells have been activated and they attack the tumour cells, the dying tumour cells will then be labelled green. So in this instance, we can capture both the direct measurement of the target cell, but also a direct measurement of the cell death of the target cell. And this assay can be run with any immune cells, so be it T cells, natural killer cells, and obviously you can alter the effector that the activator you're putting in there to get your readout. So again, we can look at T cell killing, or we can look at ADCC assays in here. And again, the images show you here the visualization, and these are stills that are taken from a video that's actually on our website. So what we're looking at here are SCOV cells, which have got a nuclear red label in them, and then we've got PBMCs in here. And if you look through the time course here, you can see there's a cell here that's actually being attacked by the T cells amongst our PBMC population, and these have been activated with anti-CD3 and IL-2. Upon the attack here on the tumour cell, you can see we eventually get this degranulation of the tumour cell, and then eventually it goes into apoptosis and we get this green marking here to show you our caspase signal. So the IncuCyte ZOOM will actually count the number of red objects in the image to give you a direct readout of the proliferation of the tumour cell, but it can also mask the green objects in here to give you a direct readout of the cell death of the tumour cell. And if we look at the graphs here, we can just see some example data where we've got activated CD3 IL-2 PBMCs, and if you look at the blue line here, you can see that the proliferation of the target cells has been slowed down and then reversed or killed. And in the absence of the activation, you can see the target cells grow as you would expect. The other graph here is looking at the green object count, and the target cells alone grow normally and there isn't any cell death. But then when you've put the activated PBMCs in there, you can see this green cell count goes up over time, showing you've got cell death. And again, we can go on to do a nice 96-well assay here, and the data here is actually showing ADCC, so antibody-dependent cell cytotoxicity. And this is looking at trastuzumab or Herceptin, and again we're looking at SKV03 cells, and you can see there's a nice concentrated related effect in the plate. And again, if we convert this data into time course analysis, here you can see the red line being the control cells and the proliferation of those over time. Here we have our positive control, which has got the CD3 and IL-2 in here, showing you nice cell lack of cell proliferation. And then we can see a nice concentrated related effect here with the trastuzumab, with the highest concentration here showing the reduction in cell proliferation. Again, we can take area under the curve and convert this into a CRC. And then the data underneath is showing the green count, so the apoptosis count. And again, we can see here we have our positive control showing us a nice cell death. But with a slightly later time course, we can see the effect of Herceptin, where we can also see a nice concentration related effect on the amount of cell death of our target cell. And again, a CRC. The last assay here on the poster is actually looking at phagocytosis. So this particular assay, we're using target cells that are macrophages that are known to phagocytose objects. And the assay that we've set up here is using the Frodo labelling So in a neutral environment, the Frodo label has no fluorescence, but once that gets internalized into the cell and inside the phagosome and the lysosome where the pH is different, we then get a nice fluorescence read. And this assay can be set up with either Frodo labeled bioparticles or Frodo labeled apoptotic cells. The images here are data shown with a mouse macrophage cell line in combination with Frodo labeled apoptotic Jurkat cells. And you can see here that there's just a nice confluent layer of cells, but this is actually a combination of the macrophage cells and the Jurkat cells. And at time 0, all you can see is the phase image. By 12 hours, you can begin to see there are red areas appearing, and this is where the macrophages have eaten the apoptotic Jurkat cells, and they are now in the lysosomes, and we're getting a release of the fluorescence. And then 24 hours, so another 12 hours on, we can see there's more red fluorescence in this image. You can also see there are areas of cell clearance here as well, and this is where the macrophages have cleared away those apoptotic Jurkat cells. The IncuCyte ZOOM can then detect where those red areas of red fluorescence are and provide a mask. And in this particular readout, we're looking at area coverage of the fluorescence within the images. So if we now convert the image analysis into a graphical form, this here is showing the same J774 cells with the apoptotic Jurkats, and this is with increasing ratios of target to effector cells. So the more apoptotic Jurkat cells we're putting in there, the more fluorescence we're seeing reading out over time. And as a control, we've got also non-apoptotic Jurkat cells that have been also labelled with the Frodo, and you can see there's a very, very small amount of fluorescence here showing that these haven't been phagocytosed. And again, we can convert this to area under the curve and have a look at the bar graph here, and this clearly shows that as the ratio increases for the apoptotic cells, we see a nice increase in the fluorescence. And again, going on and looking at pharmacology, here we're looking at a couple of different phagocytosis inhibitors and looking at concentration response curves across the plate, and you can see there's a nice concentration related effect. And here we have our controls at the bottom. Again, if we then look at cytochalasin D response and have a look at the time course, we can see there's a nice concentration related effect. We do see some level of a biphasic curve here. We see a very fast initial response, and then it flattens off and then it comes up again. And we don't really understand, we don't know the biology behind this at the moment. But again, if you convert this area under the curve, you can get a nice concentrated related effect. The last example on the bottom here is where we've done some investigations looking at Cd47 marking. So Cd47 is the eat me, don't eat me signal which appears on cells. And these particular cells that we're using, the CCRM, CEM cells, in their normal basal state have the marker on them and are labelled for not eating. But by putting the anti-CD47 antibody in there, we're blocking that marker, and now the bone marrow derived macrophages that we're looking at here see these cells as food and will eat them. And you can see here as the antibody concentration increases, we see an increase in the fluorescence. And again, we can convert this to a bar graph. So I think these three assays exemplify the use of the ZOOM in this particular field of biology. But using the system, it enables you to look at the full-time course of the biology, provides you with nice images and the possibility of time-lapse videos to give you much more insightful data. Thank you very much.